I'm going to start working through this. There's several sections here, and I really only want to get through the first two sections, which introduce you to kind of how to use the SDK to access the data. And then there's some sections below that on computing tuning curves and looking at correlations that I'm not going to try and get through today. Just I want to make it so that you can access all of the data that you're going to want to that you can get out of the NWB files. So to get set up, first we're going to import some standard packages, NumPy, Pandas, Matplotlib, and then we're going to set up the Brain Observatory. The key thing here is that there's a manifest file. If you're working on your local machine for the first time, you just actually would just get rid of this call for the manifest file, and it'll create a manifest file. And that manifest file then tells it where to download data to. And once data is downloaded, when you use the SDK in the future, it'll point to the files that already exist. So if you use this on a local machine, the first time, leave that blank. The second time, point it to where that manifest file is located. But here I've set it up with a manifest that's in this AWS environment. All right. So what I've instantiated now is this Brain Observatory cache that then looks at that manifest file and which has all of this information about the data set. And we can use a number of functions to kind of find out about the parameters of the data that we are able to access and that we'll need to use in order to kind of find specific experiments and specific data sets. And so there's some functions that kind of help us do that. So we want to start by, what are all of the areas that there's data been collected in? We call these visual areas or brain areas, we call them targeted structures. These are structures that were targeted for recording. And so we have a function called get all targeted structures. And if I run this, what it returns is a list of strings, and these strings are the names for all of the areas that we've collected data from. They all start with the prefix viz, that means visual cortex, and then they have, this is area AL, area AM. VisP is the primary visual cortex, so that's what we call V1. And so these are the 6 visual areas that we've collected data from. We have similar functions to get other information. So for instance, if we want to find the imaging depths, if we want to see what are all the options for imaging depths where we've collected data, you see there's a lot of different imaging depths that different data sets were collected from. We also used a lot of different Cree lines. So again, I think you probably can notice this pattern of get all X, get all Y. So get all Cree lines gives us a list of the Cree lines. And this is one of the things that's valuable is that, if you want to use the Cree lines, and I'll show you how we do this in a minute, to identify a particular, say you want to get data that's from the VIP interneurons, you're going to need to pass it this exact string. And so it's good to kind of have this as a reference function so that when you forget, is it iris with a lowercase r or a capital R, you can get the exact string just by using these functions. I'll make this a little bit bigger. All right, so those are all the Cree lines. We also used a couple of different reporter lines. So here we get a list of the reporter lines. So the reporter lines, that's the GCamp 6 indicator. And for most of the data, we use GCamp 6 fast. And it was either AI93 or AI148. These are different constructs of this calcium reporter. For a little bit of data, we used GCamp 6 slow, which was either AI162 or AI94. Depending on what you're doing, you might want to target the slow data, or you want to explicitly exclude the slow data and only focus on the fast data. And so you might want to use the reporter lines to select data sets based off of that. Let's look also at the different stimuli that were used. So get all stimuli gives us a list of the names of the stimuli that are used. And these stimuli, if you saw my talk at the beginning of the morning, not all stimuli are presented in each session. And so we also have this organized with different session types. And you'll get a list of the names of our different sessions types: 3 session A, 3 session B, 3 session C, and 3 session C2. And the difference between C and C2 is that partway through our data collection, we found that our locally sparse noise stimulus which had a 4.5 degree spot, stimulus spot size, wasn't very effective in the higher visual areas. And so we added in addition to that 4 degree spot, we added another part of the stimulus with or closer to 9 degree spots, but we call it 8. And so we renamed the session C2. And so some of the data will have 3 sessions C, and some of the data will have 3 sessions C2. All right, so you can see there's a lot of functions here just to kind of familiarize you with what's the parameters of the data set. And they really is just kind of this BOC get, we can get all Cree lines. We showed you that, stimuli targeted structures. There's a lot of other functions that are, if you just kind of do this tab complete that you'll see here. And hopefully most of them are self-explanatory, and then I'll get into the ones that actually give us access to the data. But you can just kind of see what these different parameters are. All right, so I'm going to now use some of these parameters to start finding some specific data sets. So you can pick any visual area or Cree line that you want. I'm just going to, for this demonstration, I'm going to look in our primary visual cortex, VisP. I'm going to look at this CUX2 Cree line. CUX2 is a dense excitatory Cree line that's concentrated in superficial layers, so layers 2, 3, and 4. So I'm just going to define these 2 variables. And now I want to find all of the experiments, all of the data sets that were collected in primary visual cortex with CUX2 Cree line. So it's another get function under the BOC, but this is called get experiment containers. And I'm going to specify that the targeted structures is the visual area that I already defined, V1, and the Cree lines is the Cree line that I already defined. All right. And so this returned a list of experiment containers, right? An experiment container, I don't know if I've defined this terminology very clearly, but an experiment container consists of 3 separate sessions. So it's the 3 sessions that we image the same group of cells. Each session is a different compartment, a different part of the stimulus. So let's just look at how many experiments we have here. There's 16 experiments for CUX2 in V1. We can take a look at, there's a bunch of information here. So I'm going to make a data frame of this list because I find that an easy way to look at it. So each row here is 1 experiment container. And then we've got a lot of information that's included here. So we've got an experiment ID. This is a unique identifier that we can use to access this container. We have information about the imaging depth. So you see for CUX2, we've imaged it at 2 different depths, 175 and 275. All of this is in V1. All of this is CUX2. That's how we selected this group of experiments. So that doesn't surprise us. It all has the same reporter line. The donor name is an identifier for the mouse. And so sometimes mice are used more than once, but not for the same imaging depth and Cree line. Outside it, like we collect data from the same group of cells 3 times, and then for that mouse, we might image a different visual area or a different imaging depth. But so you won't see the same ID for the same imaging depth, same Cree line. All right, and so this kind of gives you some information, again, that might be useful for subselecting data that you want from this. So I'm going to pick 1 experiment from this. I've already kind of pre-filled this with a container ID. This is this ID that's in this column. So I'm actually just taking the first one here. And what I want to now start accessing the data for that experiment container. And as I've mentioned several times now, there's 3 different imaging sessions. Each session has its own data file. And so we have to, if we want to work with the data, we have to identify the individual session that we want. So let's see what sessions are available. Let me scroll this for you. So now I'm going to use this function that's called get OPHIS experiments. And this is going to give us a list of experiments. And I'm going to pass it is everything freezing on me? All right, my notebook is freezing. There we go. So I'm going to pass it the experiment container ID that I already defined in the line right above that. And this gives us, this is just marked down that got messed up. let's look at the list that came back. And again, I'm going to make a data frame of these sessions. And so here we get a list of the sessions that are included in this experiment container. So again, the imaging depth is all the same. The targeted structure, the Cree line, the reporter line, those are all the same. The donor name in this case is all the same because it's from the same mouse. The differences that we see are this ID on this far end. This now is no longer the experiment container ID, but the session ID, which points us to this very specific file for the very specific session that this is. I think the other key difference that you see here is the acquisition age. So you see, actually, in this case, 3 session C was taken first, then 3 session B, and then 3 session A. So that's the order in which these data sets were collected. You can see the age of the mouse for each of those days. This little graphic just explains which session has which stimuli that you can refer to in the future. All right, so I want to get the ID for the imaging session that contains the natural scenes. And so I'm going to use, again, this get OPHYS experiments function that we used. I'm going to pass it the experiment container ID that we already chose. And now I also want to pass it a stimulus name. So I want to identify the particular session that has the natural scenes. And so this will return, again, a list. There's only one item in that list, so I want that 0th item. Let me just make this a little bit more legible for you. I want that 0th item, and what I want from it is the ID value. And this is going to be a variable that we call session ID. So let's run this. We can now just see what that session ID is. Again, it's this 9-digit string that we see again in this list up here. It's this one here from 3 session B. All right, so this session ID is what we need to be able to actually access the data for this particular experiment. So now we can kind of dig into the part where we actually start looking at real data. So I'm going to make a dataset object. And so now I'm going to use this function called get experiment data. And I'm giving it the session ID that we just identified. And this will take a little minute, especially if we're not in AWS. This might take a few minutes because that's actually where it downloads the NWB file. All right, so now we can use this dataset object to access all of the pieces of data that are in our NWB file. And so let's just start looking at what we have available to us. So we've got a max projection. And in our standard fashion, all of our functions are get this, get that. So get max projection will give you the max projection. This is an image, or it's an array, but we can look at it as an image. Max projection. So I'm just making an image of this max projection. This is a max projection of the entire imaging movie, and so it's the maximum value for each pixel. And it gives you a pretty good feel for how many cells are in the field of view. It's not perfect. There are cells that won't show up very, very strongly in the max projection, but it gives you that first pass glimpse of all of the cells that are in our field of view. We also have the segmented masks that were used to identify each cell and get all the traces. So again, using our dataset object, get ROI. It's not auto-completing for me, but it's mask array is equals. And so this is an object. Let me add a cell here. The shape of this is 174 by 512 by 512. 512 by 512 is the size of our image. And you'll see, like in this max projection, right? This is 512 by 512 pixels. And so this array, this ROI's array, it's got a single plane for each ROI that's identified. And so you can see each ROI individually. Let me just look at one of them. So that is one of the cells. We could also just look at all of them. Let's try this. That's not what I want. I want ion show. So here you can see all of the cells that were identified in this field of view. And if I leave it as an exercise for the viewer to superimpose this on the max projection and kind of see what that looks like. But this gives you all of the ROIs that were used. All right, now let's start looking at some. Let's keep going, and let's look at our fluorescence traces. The data set includes a lot of different versions of the fluorescence trace. So there's the fluorescence trace, there's the neuropil correction trace, and we also have the delta F over F, where we've computed the delta F over F across the entire session. This tends to be what I defer to. So I'm going to pull this up. The function to get the delta F over F traces returns two things. It returns timestamps and then the delta F over F array. So we can run this. So the imaging is done at 30 hertz. The timestamps are just stamps that we have kind of retroactively applied to each of the data frames. So they're just spread out at 30 hertz. I often just work with the frame numbers. All of our stimulus table information that I'll show you in a moment deals with which two-photon frame was active when, or was being collected when the stimulus began or ended or what have you. So I tend to kind of ignore these timestamps, but they are there and can be useful for certain things. So just looking at the shape of our delta F over F array, there's 174 rows. This is aligns with the number of ROIs that we saw in our mask array. And then this is the time dimension. So that's the time duration of the experiment. So let's look at some of these cells. So here I'm going to plot the first cell. So just plotting this delta F over F for the 0th item. And you see there's times when it's active and times when it's not. I'm now going to make a plot. I've pre-filled some of this just to cut down on the clicking and clacking. Here are the first 50 cells from this data set. So these aren't all of the cells, but you can see the first 50. And yeah, you can start to see that different cells are active at different times. Here's a cell, it's really nice and active, and then it kind of goes quiet for a bit and comes back. Or here's some cells that show really strong responses at different times during the experiment. So it shouldn't be a surprise why we see different cells active at different times. It really is because we're showing different stimuli at different times, and some cells respond better to some stimuli than others. So let's start looking at the stimulus information that is contained within our data set. So the first thing I want us to refer you to is something that's called the stimulus epoch table. This is really useful for stimulus epoch table. This is a useful data frame that, I don't know why this is taking a long time, that gives you a list of all the stimuli that are presented in the session and kind of the epochs when they start and when they end. So the way we did these experiments is that we would show, say 10 minutes of, here it tells us exactly, 10 minutes of static gratings, followed by 10 minutes of natural scenes, followed by 5 minutes of spontaneous activity. And then we come back and we show natural scenes again, and it's a continuation of the natural scene stimulus. And so we broke our 1 hour imaging session into blocks of around 5 to 10 minutes, where we'd show these different stimuli and then come back and keep showing more of them. And so this gives you just kind of that bird's eye view of the entire session. But we then have specific information for, and I'll show you in a moment, the stimulus table for the static ratings, so the stimulus table for the natural scenes that tells you the exact trials and when each trial began and what the parameter values of that trial were, either the grading conditions, the orientation and spatial frequency of the grading, or the image number for which image was being presented. So, but just looking at these stimulus epochs, these big broad windows of time that the different stimuli are being shown. In this cell, I'm making the same plot that I made above. I'm plotting these 50 traces of delta F over F, and now I'm just shading in the different stimulus epochs with a different color for each stimulus type. And so you can see now where we see that, these cells, like this is a cell I pointed out before, here's the cell that's really active, and then it kind of becomes quiet. And you can see that that the alignment of when it's active and when it's quiet really aligns with when we're showing these natural scenes, and then it switches to spontaneous activity and the cell stops being active. And then it comes back to more natural scenes and we see more activity. So you can start to find cells that kind of show a selectivity for different types of stimuli. And even just kind of on this course level, you can start to see some interesting things. I'll come back to this in a minute. Let's keep finding more data. So I mentioned before, in addition to the neural activity that we've recorded, we also have the kind of behavioral parameters of the animal. In this case, I'm going to look at the running speed. So again, using this function get running speed, it returns the running speed. I'm calling this dxcm. This is the change in distance. It's in centimeters per second. And then again, it also returns some timestamps. That's the second thing there. And I can plot this running speed. So you can see in this experiment, the animal has some small bouts of running early on in the experiment, but it's mostly stationary in the middle. And then there's this window kind of towards the end of the experiment, the last third of the experiment, where the mouse runs more consistently. And we can add this running speed back to the figure that I made above, where we've got the activity of these cells, we've got the shading of the stimulus, but now I'm going to add the running speed as well to that plot. And so here now you can see where we're kind of piecing all of these different pieces of information together. I think that this is a good way to kind of get a high level view of some of the things that are going on, and you can kind of dig in, and even just at this level, we can start to see some interesting things in the data. So I've picked out a couple of key cells. So that very first trace in our plot above, up here, I think is kind of an interesting one to highlight. I'm just going to replot it just by itself. And so this is the activity of that one cell. Here's our running speed. You see these epochs of stimuli. This cell is really active during the spontaneous activity and is much less active when we show the different stimuli, the static gratings, and even the natural scenes. We don't see a lot of activity. And you'll also notice there's these little gaps. They're about 30 seconds between when we finish showing one stimulus and we begin showing another stimulus. So this is probably about 30 seconds between what is the static gratings and then the natural movie that gets shown. And you'll see you see this big burst of activity there. And again, you see a big burst of activity after this epoch of natural scenes when we have this mean luminance of gray. So this is essentially the same thing as the spontaneous window. It's just shorter. And so it's not a full epoch. But so it's kind of interesting that this is a cell that's really active when there's no structured stimulus on the monitor, but as soon as we show a grating or an image or a movie, the activity gets suppressed. And this is something we see in our data set cells that do this. They're these kind of suppressed by contrast cells that I actually find really interesting. And we see some kind of differences across some of the Cree lines in terms of their prevalence. But it's not unusual to see one or two of them in every data set that we have. Here's another cell that I'm going to plot for you that I pulled out from that trace above. Again, the activity of one cell, the running speed, and then the stimulus epochs. This is a cell that seems to be pretty correlated with the mouse's running speed. You see a lot of activity that kind of aligns with when the mouse is running, and when the mouse is stationary, the cell seems pretty quiet. Here's kind of a last example of a cell that's kind of the opposite, where we see more activity from this cell, and it's probably stimulus-driven activity, but it's more active when the cell is not running. But then the responses are either smaller or just less prominent when the mouse is running. So we see kind of a bit of an anti-correlation between the cell's activity and the running speed of the mouse. So in this one experiment, just in these first 50 cells that we have up here, you can already start to dig out different types of phenomenology in the data set that we're seeing. All right, a few more things that I want to point you to. in Peter's talk, when he was doing a lot of the comparison between the calcium imaging and the neuropixels data, he talked about events. When we've done an event extraction on our data set where we take our fluorescence traces and we use an algorithm that was developed by some colleagues at UW, Sean Jewell and Daniela Witten developed an algorithm that we use to get these events. And these events have times and they have magnitudes. These aren't actually stored in the NWB file, but they are available through the SDK through the BOC. So to get these, the function isn't through the data set, it's through the BOC object. And it's get OPHYS experiment events. And again, we have to pass it our session ID. And it's going to return an array. This array has the same shape as our delta F over F array. There's 174 cells, and this is our time dimension. And so we can, again, we can plot the events. Here I'm plotting the events and the delta F over F trace for one of the cells. I've zoomed in on a particular time window. So you can kind of see what that looks like, what that event extraction did. And so we can remake that plot of the first 50 cells and the running speed using the event traces. I probably should scale this a little bit differently. It's a little harder to see, but it's the same general gist of the activity that you have there. So some analyses are a lot easier to do using the event times and a little bit cleaner to do with the event times. So that is available. But as I mentioned before, it's not in the dataset object. You have to use the BOC object to get that array, but everything else then kind of works the same way. All right, I've got a few more minutes, so I want to get to the stimulus information. We've looked at the stimulus epoch table, but like I said before, we have more specific information about the precise trials and timing of each of our stimuli. And so we're going to get the stimulus table first for the natural scenes. So using the dataset object, get stimulus table, and then we want to pass it the string for that stimulus. And this is, again, why some of those functions I showed at the beginning are really useful. So the stimulus is called natural scenes. It's not called natural scene. It's not called natural images. And you have to get that string right in order to get it. But that's one of those errors that people learn pretty quickly. And so this gives us a data frame. Let's take a look at it. I'm just going to look at the first few rows. So each row is a trial. This column called frame that tells you the ID of the image that's being shown. And I'm going to show you in a minute how you can use that ID to actually see what the image is. But this tells you which image number was shown, and then the start and the end. And the start and the end time are in the 2p intervals, frame intervals. So we can use this start and end time directly back in this figure here, and it's in that scale of imaging frames. Let's look at the stimulus table for static gratings. Just to compare, get stimulus table, static gratings. So again, looking at the first few trials, we have a little bit of different information here. So instead of just a frame number, we've got the parameters of the gratings that are shown, the orientation of the grating, the spatial frequency of the grating, and the phase of the grating. So we have 4 different phases that we use when we flash these images. And then again, the start and the end time. So the tables look different for the different stimuli based on what the parameters of those different stimuli are. But kind of all of this key information is here. And I mentioned that you can use this ID, this frame number, to identify which image is actually being shown for the natural scenes. And this is available in here in what's called the stimulus template. So again, using dataset, get stimulus template. And we still want to tell it natural scenes. So we have templates for the natural scenes and the natural movies. These are the stimuli where we actually just showed images and movies from an array as opposed to things that were generated by the stimulus program. So, so taking a look at this array. So this is an array, it's shaped 118 by 900 by 1200-ish. So these are the 118 are the different images that we show. And so we can look at one of these images. I'm just going to pick one of the numbers. Let's take this first one here, the number 81, the one that was shown in that first trial. And I'm going to put this in grayscale because that's how it was shown. So the very first trial of this experiment of the natural scene stimulus was this picture of a shadow against a fence. And let's see, the second trial, you can also do this programmatically rather than typing in numbers. But the second trial was this cute picture of like puppies or wolves or whatever that is. So you can see that we have a lot of different natural images. You can, if you just want to see what the images are, actually the website is really useful for just flipping through them real fast. But you can access them all programmatically here and incorporate that into your code or into your analysis. And so I'm just gonna, we can take this information. So we want to look at the plot I made above of all the activity of those 50 cells. And now I'm just going to highlight whenever that first scene was shown, that we used in the first trial, the one of the shadow on the fence. And so I didn't define something. Oh, because I had given it a scene number. I kind of skipped a step above, but so yeah, so these are all of the times when we showed that first image of the fence with the shadow on it that you can see. So this falls into the 3 different epochs when we showed natural scenes. The epochs aren't the same. They're not even the same across the different experiments, which was maybe a mistake we made. But you can see all of the times when this is shown, and if you want, you can go in more closely and pull out a single cell and take the start and end of each of these trials and look at the activity of the cell during each of those trials and start doing some statistics and analysis. All right, in the last 2 minutes, I want to just show you a couple of key things. One is the metadata that comes along with in the experiment. So get metadata. This is a dictionary that gets returned that tells you a lot of information. Some of it you already know because you used it to identify the experiment. Other pieces, the only weird place you find it is in this metadata dictionary. So the sex of the mouse, the targeted structure, we already knew that. These ID numbers, we'd already figured those out. We also tell you here the time and date at which the experiment was collected, and we've had people do some analysis looking to see if there might be circadian effects on some of the responses that could use that. The age of the animal. There's also a device name. This is which exact microscope we collected the data on. We have 5 different microscopes that were used, and we've done some analyses comparing some of the activity across microscopes, making sure that we're not seeing differences based off of the different machines. So there's some nitty-gritty information in there that some of it might not be particularly relevant, but is good to be able to access. All right, I'm gonna just jump ahead so that I want to show one little thing before I turn it over to Josh. And this is how you can access the calcium movies. And again, you can only do this when you're in AWS, either in our portal or if you're accessing it through AWS through your own AWS account. But we have the raw calcium movies stored in AWS. And so this code here, you're pointing it to a particular directory where we have these OFIS movies. And you can get the raw data path for a particular session. We've already identified our session ID. And this will create the path. And then you can access this data. And let's just plot it here. It's supposed to be animated, but maybe something's maybe not fully installed. But you can see this is the calcium movie that we just pulled up. And you can, if you're somebody who's interested in kind of how we do the cell segmentation, this is an area of active research in the field. We've got, 1,400, 1,500 hours of imaging data that is really rich for kind of working on those things. And there have been a lot of people that have used the data in this way. And I just want to make sure everybody sees how they can do that. If AWS isn't your thing, the other thing you can do is send us a hard drive and we can put movies on it and send it back to you. But really, you should get up to speed and use AWS for this because it's just a lot easier. Okay, At this, I'm going to wrap up and turn it over to Josh so he can tell you about accessing the NeuroPixels data.